Hello, we're glad you've joined us for this live webinar, Advanced Assays for High-Throughput Assessment of Ion Channel Inhibition and Cardiac Toxicity. I am Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Molecular Devices. Molecular Devices is one of the world's leading providers of instruments, software, and reagents for life science research, pharmaceutical, and biotherapeutic development. With over 130,000 placements in laboratories around the world, the company's instruments have catalyzed brilliant scientific research described in over 25,000 peer-reviewed publications. Included within a broad product portfolio are platforms for high-throughput screening, genomic and cellular analysis, colony selection, and microplate detection. These leading-edge products empower scientists to improve productivity and effectiveness, ultimately accelerating research and the discovery of new therapeutics. To learn more, visit www.moleculardevices.com. Let's get started. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. You can post questions to the speakers during the presentation while they're fresh in your mind. To do so, simply type them into the Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at lower left. I now present today's speakers, Zou, PhD, and Zou Sirenko, PhD, both from Molecular Devices. Dr. Zou is a field applications scientist supporting reagents, Flex Station 3, FLIPR, and automated electrophysiology platform. She received a PhD in biology at Pennsylvania State University and completed postdoctoral training in the Department of Neuroscience at Johns Hopkins University School of Medicine. Dr. Zou has 18 years of experience in molecular biology and genetics, 8 years of experience in ion channel biology, 4 years of experience in cellular imaging, and 8 years of experience in assay development and high-throughput screen. The latter includes 5 years of experience as a project leader at the Johns Hopkins Ion Channel Center. Dr. Zou has authored and co-authored in 20 peer-reviewed papers in prestigious journals, including PNAS, Cell, Journal of Neuroscience, Molecular Pharmacology, and Assay Drug Development Technologies. Dr. Sirenko is a research scientist working on development of new technologies for imaging and high-content analysis. She also has previous experience working on drug discovery and drug development at several biotech companies in the San Francisco Bay Area. The focus of Dr. Sirenko's current scientific research is on development methods for high-throughput in vitro toxicity screening, including 3D models. She has a PhD in biochemistry and BS in biology. Dr. Zou will now begin her presentation.
Good morning. Today I'm going to talk with you about predictive assessment of cardiac safety using FLIPR Tetra Plus, high-throughput screening assays for in vitro potency evaluation of cardiac ion channel inhibitors. Cardiac safety is of paramount importance in contemporary drug development. As you can see from this slide, 95 compounds were withdrawn from the U.S. market due to their unwanted side effect. Cardiac vascular safety is one of the leading reasons for the discontinuation of these drugs. From 1990 to 2010, 16 marketed drugs were withdrawn due to arrhythmia. These drugs are listed in this table. As you can see, 14 of them were withdrawn after 1997. These drugs include compounds seeking to rectify cardiovascular problems, for example, anti-arrhythmia agent Aconite, but also includes compounds with totally different indication, for example, antihistamine, tafenidine, and estamazole. These compounds cause QT prolongation and increase risk of toxadipone, sometimes even cause sudden death. Electrical activity of heart is measured by ECG or electrocardiogram, by placing electrodes on specific points on skin to measure the activity. In ECG, the interval between Q wave and T wave is called QT interval. QT interval corresponds to ventricular depolarization and repolarization. This figure shows recording of cardiomyocytes. This is a cell, and this is a glass pipette. One beat corresponds to one action potential. Ventricular action potential duration corresponds to QT interval. There are 5 phases in ventricular action potential. A variety of channels play important roles in generation of ventricular action potential. For example, potassium channels, they play important roles in phase 3 for repolarization. If repolarization is slowed, the duration of ventricular action potential is prolonged. Correspondingly, QT interval is prolonged. Sometimes, prolongation will cause toxicity. hERG channel is included by human esophageal gene. It is a voltage-gated potassium channel expressed in cardiomyocyte. It plays a very important role in repolarization. As shown in this slide, blue represents normal hERG current. If the channel activity is blocked by compounds, its current will be greatly reduced, as shown in red. As a result, repolarization is slowed, and duration of ventricular action potential is prolonged. It causes long QT syndrome. hERG has a very unique feature. It shows promiscuous interactions with compounds of various chemical structures. In fact, among these drugs, withdrawal due to their unwanted side effects. Many of them inhibit hERG. Given the importance of cardiac safety, FDA has provided guidelines for cardiac vascular risk assessment of new chemical entities. One is ICH S7B, which is non-clinical evaluation of the potential for delayed ventricular repolarization, QT interval prolongation by human pharmaceuticals. And the other one is ICH E14, which is a clinical evaluation of QT and corrected QT interval prolongation and pro-arrhythmic potential for non-antiarrhythmic drugs. Since the most common mechanism of QT interval prolongation by pharmaceuticals is inhibition of hERG, that is responsible for IKR, it is required in ICH S7B to assess compound effects on hERG. This figure shows the general non-clinical testing strategy for assessing risk for delayed ventricular repolarization and QT interval prolongation. In this strategy, compound effects will be evaluated on IKR or hERG. Results from these assays will be used together with other information to provide integrated risk assessment of compounds. Several assays have been developed to evaluate compound adverse effects on hERG or cardiomyocytes. These assays include fluorescence-based serum influx assay using FLIPR and automated electrophysiology assay of hERG. Both assays are performed using cell lines stably expressing hERG channels. Compared to automated electrophysiology assay, throughput is higher and the cost per data point is lower in FLIPR serum influx assay. In the following few slides, I'm going to show you the data from FLIPR serum influx assay and the comparison between the results from FLIPR serum influx assay and automated electrophysiology assay. I would like to mention that FLIPR Tetra System can be used to measure calcium dynamics in cardiomyocytes differentiated from iPSC. My colleague Oksana will show you the data from FLIPR calcium assay of cardiomyocytes in her presentation. Before showing you the data, I would like to provide you an overview of FLIPR Tetra System. FLIPR Tetra System, shown on the left, is the latest generation of FLIPR system. It is a bottom-read fluorescence imaging plate reader with integrated liquid handling. It can read and dispense simultaneously, which is required for fast kinetic assays, such as calcium assays and thallium influx assays. FLIPR can be applied to 96, 384, or 1536 well formats. It utilizes LEDs as excitation resource and uses EMCCD or intensified CCD camera to detect signals. Optics is user-changeable. LEDs and filters of various wavelengths are available for assays using different dyes of fluorescence proteins. There are 5 stages on FLIPR. 4 of them are for plates, which allows multi-addition assays. The system is operated via ScreenWorks software. ScreenWorks is industry-renowned software. FLIPR protocols are set up via drag and drop of pre-configured modules. ScreenWorks provides on-the-fly data analysis. If users want to apply different software for data analysis, data can be exported in worksheet-compatible format to be used in other softwares. This slide shows traces from FLIPR's thallium influx assay of hERG channels using a stable cell line. The x-axis is time. The y-axis is response over baseline. Green traces are from these wells where hERG channels are activated to open. Black traces are from these wells where the activity of hERG channels is completely blocked. One trace is from one well. In this experiment, assay plate was detected on FLIPR for 10 seconds to establish fluorescence baseline. Then, stimulus buffer containing both thallium ions and potassium ions was applied to all the wells. Fluorescence signals were detected for another 120 seconds. As you can see in this slide, fluorescence signal increased dramatically in wells where hERG channels are activated to open. However, in these wells where the activity of hERG is completely blocked, the increase of fluorescence signal is much smaller. The difference between the two populations reflects activity of hERG channels, which is about 2-fold increase in fluorescence signal. Data shown in the previous slide was obtained using the new FLIPR potassium kit. This kit utilizes a novel thallium-sensitive dye and a special masking dye. The thallium-sensitive dye is loaded into cells. Once potassium ion channels are activated, thallium ions flux into cells and bind to thallium-sensitive dye. As a result, fluorescence signals increase. This assay allows functional measurement of potassium channel activity. Unlike thallium-sensitive dye, masking dye is in the extracellular solution and can significantly reduce the background. The assay is homogeneous and non-wash assay. In such assay, well variations are greatly reduced by eliminating washing step, and Z prime factors are increased. FLIPR potassium kit is very easy to use. Component A, B, and C are mixed together to make dye loading solution. Add dye loading solution to microplates and incubate for 1 hour at 37 degrees. Then the plates are ready for assay on FLIPR Tetra Plus. hERG channels are voltage-gated potassium channels. They can be activated to open by membrane depolarization. In assays, we increase extracellular potassium ion concentrations to depolarize membrane to activate hERG channels. To obtain optimal results, assay optimization is required. In hERG thallium assay, we tested various concentrations of thallium and potassium ions to identify an optimal condition. hERG channels were activated by 10 millimolar or 20 millimolar extracellular potassium ions. Fluorescence signals were measured when the extracellular thallium ion concentration was at 0.5 millimolar, 1 millimolar, or 2 millimolar. Blue lines are from the experiment where channels are activated by 20 millimolar potassium ions in extracellular solution. Light green lines are from the experiments where channels were activated by 10 millimolar potassium ions in extracellular solution. Orange lines are controls, where hERG channels are not activated. We found the fluorescence signal window is the largest, with stimulus buffer of 1 millimolar thallium and 10 millimolar potassium. Then we tested compound effects using this condition. Thallium assay has been previously developed using BRAND-F kit. BRAND-F kit requires washing. To assess the assay performance, we tested non-hERG inhibitors, astamazole, pimozide, and tafenidine using FLIPR potassium kit and BRAND-F kit. We compared assay window, Z prime factor, and compound pharmacology from both kits. These three were from experiments using FLIPR potassium kit. These three were from experiments using BRAND-F kit. As you can see, fluorescence signals of this group increased approximately twofold. Fluorescence signals of this group increased approximately 0.5-fold. FLIPR potassium kit allows larger signal window. Z prime factor of FLIPR potassium kit is 0.85. Z prime factor of BRAND-F kit is 0.64. IC50s of testing compounds are very similar between the two assays. According to data, FLIPR potassium kit can be used to assess compound effects on hERG. The benefit to use FLIPR potassium kit includes larger signal window and a better Z prime factor. Electrophysiology is gold standard to measure ion channel activity. To further assess whether FLIPR thallium assay can be used to predict compound adverse effects on hERG, we tested 7 non-hERG inhibitors and compared their IC50s with values from automated electrophysiology assay using Ironworks Barracuda. Concentration-dependent curves from these two assays were plotted in the top left and top right panels, respectively. IC50s are summarized in the table below. Ratios were taken between IC50s from FLIPR assay and IC50s from Ironworks Barracuda assay. As you can see, all ratios are less than 3, suggesting that compound potency is similar. In addition, the compound rank order retains. Therefore, FLIPR thallium assay can be used to predict compound effects on hERG. Given the low cost and higher throughput, FLIPR thallium assay can be used in early drug development stage to assess compound effect. Although the most common mechanism of QT interval prolongation by drugs is inhibition of hERG, prolongation of the action potential can result from decreased inactivation of the inward sodium or calcium currents. NaV1.5 is a voltage-gated sodium channels expressed in cardiomyocytes. In phase 0, the upstroke of the action potential is primarily a consequence of a rapid transient influx of sodium currents through NaV1.5 channels. CAV1.2 is L-type voltage-gated calcium channels expressed in cardiomyocytes. In phase 2, the plateau of the action potential is a reflection of a balance between the influx of calcium currents through CAV1.2 and outward repolarizing potassium currents. FLIPR assays have been developed to assess compound effects on NaV1.5 and CAV1.2 channels. FLIPR assays of NaV1.5 and CAV1.2 channels are using membrane potential dye and FLIPR calcium assay kits, respectively. As you might know, membrane potential dye kit is a primary assay kit for monitoring ion channel activity. It measures membrane activity regardless of ion types. It is a homogeneous assay. Z prime factors are increased by eliminating washing errors. Molecular devices also offer several FLIPR calcium assay kits, including early tox cardiac toxicity kit, formulated for applications in cardiomyocytes. These calcium assay kits are established reagents for calcium mobilization screenings. They provide larger signal window and are less toxic. In the following slides, I'm going to show you the results from FLIPR membrane potential dye assay of NAV 1.5 channels. This slide showed data from CHO cells expressing potassium channels. Elevated extracellular potassium ion concentrations were used to depolarize the membrane. Membrane potential was measured using both membrane potential dye and manual patch clamp. Comparison of the data with patch clamping results showed good correlation. It's demonstrated that membrane potential dye can measure membrane activity. Membrane potential dye has fast response time. This slide shows an overlay of data from manual patch clamp and FLIPR membrane potential dye assay. 60 millimolar potassium chloride was used to depolarize the membrane. Orange triangle represents data from FLIPR membrane potential dye assay, and green diamond represents data from manual patch clamp. As you can see, membrane potential assay data and patch clamping results show good correlation. Veratridine is a known sodium channel opener in membrane potential fluorescence assays of NAV 1.5. Veratridine is used to hold the sodium channel in its open state. Rapid influx of sodium ion into the cell depolarizes the membrane, leading to an increase in fluorescence. There are two FLIPR membrane potential assay kits, blue and red. As shown in the slide, comparison of the cellular response to veratridine using the FLIPR membrane potential red and blue assay kits is similar. EC50 concentration using the blue assay kit is slightly right-shifted at 37.2 micromolar compared to 20.4 micromolar with the red assay kit. Tetrodotoxin is a potent sodium channel blocker. Its effect on NAV 1.5 was evaluated using both red and blue assay kits. Results showed that IC50 was 0.79 micromolar with red assay kit and was 2 micromolar with blue assay kit. Both are similar to published conventional patch clamp data, 2.4 micromolar. Change in fluorescence was greater with red assay kit. In addition, Z factor of red assay kit was higher than that of blue assay kit. To obtain optimal results, we recommend comparing these two kits in your assay. Lidocaine is a local anesthetic that works by blocking sodium channels in a user-dependent manner. Results of the FLIPR membrane potential assay kit showed that IC50 was 9.55 micromolar with red assay kit and was 51.3 micromolar with blue assay kit. Both are more potent than the results from electrophysiology data. Z factor is higher with red assay kit. Higher potency measurement in this assay should be expected, as veratridine locks the channel in the open state, maximizing the exposure of the channel to lidocaine and thus increasing its apparent potency. Next, I'm going to show you some data from FLIPR assay of CAV 1.2 channels. In the FLIPR calcium channel assay, elevated potassium ion concentrations were used to evoke the response. To find optimal conditions, response was evoked by potassium chloride at various concentrations. Representative traces in response to various potassium concentrations are shown in the left panel. Signal window versus extracellular potassium concentration was plotted in the right panel. We found 20 millimolar potassium chloride resulted in larger signal window. Then we tested non-inhibitors using this condition. Two known L-type calcium channel inhibitors, nifedipine and rapamycin, were tested. Concentration response curves were plotted. IC50 of nifedipine was 42 nanomolar, and IC50 of rapamycin was 10.4 micromolar. Both are in good agreement with results from electrophysiology assays, suggesting that this FLIPR assay can be used to predict compound effects on CAV 1.2 channels. In summary, cell-based fluorescence functional assays have been developed using the FLIPR Tetra Plus instrument for in vitro potency evaluation of inhibitors of hERG, NAV 1.5, and CAV 1.2, 3 ion channels, playing important roles in generating and maintaining normal cardiac electrical activity. Compared to results from electrophysiological recording techniques, compound potency of non-hERG inhibitors, tetrodotoxin, and non-calcium channel inhibitors from FLIPR assays is comparable. In addition, compound rank order retains. Given its low cost and higher throughput, these FLIPR assays can be used for in vitro compound assessment at early drug development stage. Thank you very much for your attention.
Thank you, Dr. Zou, for that informative presentation. We will now hear from Dr. Sirenko. I will hand the presentation over to her.
Thank you for your introduction. In the second part of our webinar, I will describe phenotypic assays that use induced pluripotent stem cell-derived cardiomyocytes for evaluating the effects of compounds on cardiac physiology. Cardiotoxicity remains one of the main reasons, comparable with hepatotoxicity, for drug attrition during clinical trials. Current preclinical approaches for cardiophysiologic evaluation of chemicals rely almost exclusively on animal models, and this approach has significant limitation in terms of the cost and complexity of the studies. So there is a great pressing need for the development of comprehensive multiparametric screening strategies that provide improved predictability of cardiotoxic effects. Novel approaches need to rely on functional studies instead of traditional cytotoxicity measurements, because interference with normal cellular electrophysiology can have impact on organ function. As an alternative to primary cardiomyocytes and myocardial tissue preparations, human inducible pluripotent stem cell-derived models have recently emerged as physiologically relevant and highly reproducible options for cardiotoxicity testing. iPSC-derived cardiomyocytes especially attractive as they form synchronously beating monolayer that can be used to reproduce drug-associated physiologic phenotypes. We used fast kinetic fluorescence assay that monitors changes in intracellular calcium fluxes. Fluorescent measurements of intracellular calcium flux were performed using FLIPR Tetra high-throughput screening system from molecular devices. The assay can be done in 96 or 384 well formats with automatic addition of compounds. We have tested a library of cardiotoxic compounds that contains 131 compounds, including known anti-arrhythmia, anti-cancer, anti-inflammatory, neuroleptic compounds, as well as cardio-safe drugs used as negative controls. Compounds represented different mechanisms of action: blocking of ion channels, adrenal receptor antagonists, kinase inhibitors, and others. Several phenotypic descriptors were derived using ScreenWork Peak Pro software. The readouts included peak frequency, peak spacing, amplitude, peak width, as well as peak rise and decay times. The assay allowed detection of various phenotypic responses, including increase and decrease of peak amplitude and frequency, prolongation of a repolarization step resulting in increase of decay time and peak width, which was observed with known hERG blockers. Also, peak irregularities and increase of distance between peaks is seen as a result of the effect of sodium channel blockers. Evaluation of changes in beating profiles is important for detection of functional effects, but high-content imaging is an essential complementary assay for monitoring morphological changes and cytotoxicity effects of compounds. Calcium AM was used to identify viable, metabolically active cells and to define morphology features. Cell-permeable nuclear dye hERG was used to measure total cell count and assess nuclear shape. MitoTracker Orange was used for detection of cells with intact mitochondria. Based on testing of library of cardiotoxic compounds, we have estimated the assay sensitivity is greater than 80% with specificity greater than 90%. Concentration-dependent effects of screening compounds on peak width and beating rate were visualized using hierarchical clustering and heat maps. Analysis allowed clustering of similar classes of compounds together that may provide insight for the mechanism of toxic effects. The method allows to address several major barriers that preclude wide use of human stem cell-derived cells as models for cardiotoxicity screening, which include throughput, data integration across multiple parameters, and quantitative assessment of concentration responses. The effective concentration of 50% of maximum response, EC50s, is a reference point that is used most frequently in the analysis of in vitro data. However, the use of AC50 may be problematic in cases when 50% response is far outside of the range of concentrations tested. For that purpose, using benchmark concentration or an effective concentration that causes deviation of the value from the control for more than either 1 or 3 standard deviation overcomes the limitation of AC50 analysis. Benchmark concentration is used for toxicity evaluation and can be used for ranking compounds according to their toxicity effects. In the collaborative project with the National Institute of Environmental Health, we've used the described assay to test number of environmental chemicals for potential cardiotoxicity effects. It is estimated that more than 20% of all cardiovascular diseases related to environmental exposures. In our work, we evaluated feasibility of combinatorial screening approach for analyzing functional and mechanistic cardiotoxicity. We used calcium flux measurements in combination with high-content imaging to evaluate concentration-dependent effects on cardiomyocyte physiology, mitochondrial membrane potential, and cell viability. The library used in these studies is comprised of 69 unique chemicals with mostly limited or unknown evidence of cardiotoxicity. The library included different classes of compounds: pesticide, flame retardants, polyaromatic hydrocarbons, and some cardiotoxic drugs. Shown here are representative phenotypic responses, which include prolongation of repolarization step, as well as changes in amplitude and frequency. Cluster analysis of quantitative concentration response data for cardiomyocyte peak frequency and cell viability displayed here. Compounds that had most of the effect are shown in additional panel. This figure represents the ranking of 52 selective chemicals. Selective means that the effect of chemicals have a ratio of effective concentration for viability to calcium flux phenotype greater than 3. Data presented for 30 minutes and 24 hours of exposure. Most selective compounds shown on the top. Effect in concentration values presented for 69 unique chemicals assessed for 18 different phenotypes. Data was integrated in ToxicPy software for quantitative bioactivity profiling. Plots indicate relative chemical rankings per chemical class. ToxicPy profiles for selected representative chemicals indicated. In the second part of my talk, I will describe 3D models for cardiotoxicity. 3D cultures believed to have advantage of closely recapitulating aspects of human tissues, including architecture, cell organization, and cell-to-cell interactions. In the present study, we developed and characterized kinetic and confocal imaging methods compatible with automated instruments and show utility of 3D cardiac spheroid cultures for toxicity screening. Cardiac spheroids spontaneously contracting and their movement can be observed in transmitted light. Fluctuation in fluorescence intensity consistent with mechanical contractions were observed after treatment of spheroids with calcium dyes. Images were taken by time-lapse acquisition for spheroids stained with FLIPR calcium-6 dye and treated with different compounds. Fluorescent measurements of intracellular calcium flux were performed using FLIPR Tetra High-throughput screening system or imaging system IXMC. The workflow for the screening assay is presented on a slide. Human stem cell-derived cardiomyocytes were cultured in ultra-low attachment plates to initiate 3D spheroid formation. U-shaped plates were used for spheroid formation, compound addition, calcium flux assay, and confocal imaging. We developed and characterized a protocol for image acquisition and peak analysis using the ImageExpress micro instrument. The recent version of MetaExpress software contains an addition of peak analysis algorithms similar to ScreenWorks Peak Pro analysis. The screenshot presented here shows the user interface for analysis setup and calculated readouts. Calcium flux assay on a FLIPR Tetra system with spheroids performed similarly to standard 2D culture assay. Similar acquisition and analysis protocols were used, but performance of the assay was dependent on centering spheroids in the well and also size of spheroid. Phenotypic responses were in good agreement with data from 2D cultures tested in parallel. Specifically, it was observed blockage of beating with digoxin, increase of spacing between peaks by lidocaine, increase of beating rate and amplitude with isoproterenol, as well as decrease of peak amplitude with propranolol. But also, there were several important differences in the beating profiles between 3D spheroids and 2D cultures. First, the beating rates were significantly higher for 3D spheroid cultures in comparison to 2D cultures, plated at the same seeding density into conventional 384 Corning plates. Second, the observed amplitude of fluorescence spikes was significantly lower for 3D spheroid cultures. Third, the widening of peaks and increase of peak decay times were less profound for 3D spheroid culture in comparison with 2D culture. And finally, there were clearly different concentration and time responses for number of tested compounds. Interestingly, in 3D culture, it was apparent that concentration dependencies and EC50 values were shifted toward greater concentration, suggesting that the same dose of compounds were having less of the effect in 3D spheroids in comparison to 2D cultures, and ranking number of compounds, notably hERG inhibitors, as substantially less toxic. It is possible that penetration of those compounds into cardiac cells, as well as diffusion inside spheroids, may be limited and delayed in the case of 3D cultures. Out of 14 cardiotoxic compounds tested, 4 compounds, including doxorubicin, staurosporine, astemizole, and sunitinib, caused significant changes in spheroid morphology and cell viability. Spheroids lost their spherical shape, appear disintegrated, or become flattened. In addition, the number of viable or calcine AM positive cells decreased. Cytotoxicity effects for the same compounds were also observed for 2D cultures. Images were taken using automated confocal microscope with a large field of view camera. The system captured spheroids in a single image using 10x objective with either 11 images at 10 micron increments or 17 images at 5 micron increments. 3D analysis of the images was done using the features of MetaExpress software, which enabled conversion of stack of 2D images obtained by confocal acquisition into 3D space with appropriate detection and segmentation of objects. 3D analysis allowed for measuring several parameters important for phenotypic characterization of drug-induced cytotoxicity, including spheroid diameter, size, volume, as well as the numbers and intensities of live or dead cells. EC50 values in micromolars were measured for tested compounds using peak count as a readout. Some compounds had comparable EC50s. A number of other compounds had significantly higher EC50 values for 3D spheroid cultures, more prominent at earlier time points. In a summary, we described here advanced method for imaging and analysis that allow quantitative characterization of complex phenotypic effects. And second, 2D and 3D cardiac cell models from stem cell-derived cells can be successfully used for toxicity evaluation and compound screening. Thank you so much for your attention. I would like to acknowledge Carol Crittenden, Matthew Hammer, Sean Keating from Molecular Devices, Michael Hancock, Kobe Carlson, and Blake Anson from Cellular Dynamics, Fabian Grimm and Ivan Rusin from Texas A&M, as well as Kristen Ryan and Raymond Tice from National Institute of Environmental Health Sciences. Thank you.
Thank you, Dr. Zou and Dr. Sirenko, for your presentations. A quick before we begin the question and answer session, I want to advise that a series of poll questions will appear on your screen. You may select your answers, then close the poll questions by clicking on the X in the right corner. A quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. Dr. Zou and Dr. Sirenko will answer as many questions as time permits. And the first question is, the results of the hERG thallium assay are very good. Can we use the assay, can we use the kit for other targets, and what targets have the kit been applied to?
Yes, the kit can be used to other targets. In theory, the kit can be used to any targets that allow the thallium flexing to the cells. We have app notes of potassium chloride transport, as well as voltage-gated potassium channel KV1.3 assay using this kit. And these are on our website. And besides, this kit can be also used for to look for channel openers. We do have a webinar on our website looking for there's the assay using the same kit to look for openers of another voltage-gated potassium channel.
Next one is, we have a hERG stable cell line and are interested in using your kit. Can we use the same condition that was used in your assay?
Well, our assay condition can serve as a starting point for your assay. As a good practice, to find the best conditions for your assay, we recommend that you should optimize assay conditions for your cell line and your assay.
Can we do a comparison of peak count analysis between FLIPR and image express micro? And if so, does it give the same results? And let me know if you need me to repeat. I can, I'm not sure if we're having a momentary technical difficulty, so hold on with us for just a moment, and I can repeat that question in just a moment as Dr. Zou and Dr. Sirenko are ready to answer it. But we may have just a brief blip, brief technical difficulty. Okay, are we? So let me repeat that question. Can we do a comparison of peak count analysis between FLIPR and image express micro? And if so, does it give the same results?
Yes, actually, we have done comparison with from between FLIPR and IXM, and actually we get the same count with the same sample on both instruments, even so that software was slightly different. The only difference was in standard deviation of the data, because since on image express it's less time for acquisition and you have less data point, the experimental error was somewhat greater.
Okay. Now, where is RFU? Let's see. Why? Let me just. Let's see. How is toxicity determined? Is it by peak area or RFU with comparison to the control?
So the parameter which we use for determining toxicity and also IC50 and effective concentration, we use a number of beats per minute or beats per recorded time. So it was kind of frequency of peaks.
And why is the RFU greater in 2D?
So this is something which is most likely related to biology and physiology of cells and some of the differences which we see between 2D and 3D. So one of the reasons could be that cells kind of more strained in 2D, and kind of because of that the frequency goes less, and in 3D they kind of contract more in synchronous, but it's kind of speculation, and it definitely reflects some differences between two systems, but why is that? It needs to be determined.
Okay, and as we're waiting for more questions to come in, I would like to once again thank Dr. Zou and Dr. Sirenko for their presentations. Do you have any final comments?
And thank you so much for opportunity to present. It was, yeah, nice opportunity.
Well, thank you both. And there is another question I'd just like to pose to you. What is the nature of this cell line? It's actually a two-parter. What is the nature of the cell line? Are they continuous primary cell line cultures, because it was said it is derived from the stem cell?
Yes, this is actually stem cell derived cell type. So this is one of a product of Cellular Dynamics International, but some other companies also kind of produce similar products. So those cells are derived from primary cells. Usually it's from blood cells, and by transaction of number of genes which make cells induced pluripotent. And then those cells are differentiated toward cardiomyocytes. So those cells are primary. You cannot expand them in a culture, but you can grow them for your experiments and use it and buy more from a company who produces it.
Thank you. I've got another question for you. With the FLIPR Tetra System, can we observe the cytokine effect on cardiomyocyte and channel activity?
I would say this would be kind of interesting question to address, but we haven't done that. It wasn't in scope of our studies, so we were focused on compounds and kind of known modulators of cardiac activity.
Okay, well, thank you again, Dr. Zou and Dr. Sirenko. I'd like to thank the audience for joining us today and for their interesting questions. We would also like to thank our sponsor, Molecular Devices, for underwriting today's educational webcast. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. This webcast can be viewed on demand through November 9th, 2017. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who have missed today's live event. Until next time, goodbye.